Marcia said I should not call it a crash course on the X and the Y chromosomes. So I'm going to give you a primer on the X and Y chromosomes, but especially from an evolutionary perspective. But and as Mike said, I'm going to give sort of two lectures. I'm going to start with the shorter one, which is a little bit more historically focused, and I'll tell you about the beginnings of the X and the Y. But I actually want to devote my lectures today. I want to dedicate them to two people. The first person is, I'm shown here with, this is a photo from yesterday afternoon when I was visiting my mentor, David Botstein, who is a legend in the realm of genetics and who I had the pleasure of working with many years ago. And just for to ground things here at Stanford, David was the chair of the Department of Genetics, I think approximately in the 1990s. And just a little bit of history here. David actually, together with three other colleagues, including Ron Davis here at Stanford, wrote this paper in 1980, which was proposing the construction of a genetic linkage map of the human genome. And I think this was, in fact, the first sort of white paper, if you will, proposing what would become the Human Genome Project. And I had the pleasure of starting to work with David and actually Ray White in the summer of 1979, the year before, as a wayward medical student. And thereby became the first student anywhere in the world, I think, to become the first student anywhere in the world to work on what would become the Human Genome Project. And I took up this project of making a linkage map of the human genome because, as students do, I said, oh, sure, I'll do that. Why not, right? And it took a few more years and a few additional people, but anyway, I just wanted to ground things in this place and with David, who I had the pleasure to visit yesterday afternoon. The other person I want to honor today also has very strong Stanford connections, and that is the person shown in the center here, Britt Jepson-Darbeloff. Actually, it turns out two of her grandsons are undergrads here at Stanford and are in the second row, right over there. And anyway, Britt was the first woman to receive a degree in mechanical engineering at Stanford. Just want to let that sink in. Right. Then she moved to MIT, and she was the only, to get a master's degree, she was the only woman at MIT in the Department of Mechanical Engineering. She went on to, from there, to work in various pursuits. She contributed to the design and construction of the Redstone rocket. She was at DEC, at Digital Equipment, and then at Teradyne. And subsequently, she has been an enormous supporter of women in STEM. And she and her family have been the single most generous supporter of the work in my laboratory. And so all of this is very much grounded. What I'm telling you is really a story of connections between MIT and Stanford. And so I wanted to bring these things because people make a difference. So thank you. And thanks to her grandsons for being here today. Right. Okay, so, and actually, it's funny, I'm going to come back to some of these other individuals in this photograph in my second lecture. So, but let's turn to my favorite pair of chromosomes. So, on the left, stately and statuesque, proud, the X chromosome. And to its right, with its head down, the demure and diminutive Y chromosome. Now, the truth be told, I have spent most of my career, and I'm looking at Howard here, you know this, I spent most of my career defending the honor of the little puny one in the face of many insults to its character and its future prospects, which many have considered to be quite bleak. I will not address the topic of the disappearing Y chromosome today unless you, I don't know, whatever. I'll try to avoid that subject. But what I'm going to tell you eventually is that though I have been quite fixated, my lab and I have been quite fixated on the Y chromosome for a long time, in recent years, my interest has turned dramatically to its more stately and statuesque partner, the X chromosome, especially, as I will explain, in its so-called inactive form. But so let's get then to a brief history of the human X and Y chromosomes. What I want to tell you is that, and I checked out a bunch of textbooks. I did some Google AI searches on what I should know about the X and the Y chromosomes, and they basically don't say anything about evolution. So I'm going to tell you that the only real way to understand the genetics of sex and to understand the X and the Y chromosomes is from an evolutionary perspective. So let's go there. And here I'm quoting Dobzhansky, the famous quote, "Nothing in biology, nothing in medicine makes sense except in the light of evolution." So let's go there. All right, we're going to go back to basics. How old is sex? Let's see. Maybe, okay, we'll start with easier questions. How old is the universe? This is going to be interactive, so you need to shout out some answers. How old is the universe? 13, 14 billion years. That's a good estimate. How old is the planet? How old is our planet? 4.5 billion. I hear somebody said 7,000 years, but 4.5 billion, right? Okay. So, so if the planet is 4.5 billion years old, then how old is sex? Okay, I'm going to suggest in the evolution of sexual reproduction, there were 4 great inventions. And the first invention I will attribute to, I'll credit prokaryotes with having come up with some fairly systematized way of exchanging hereditary information. And this is on the order of 3 to 4 billion years ago. So most of the age of the planet. But in prokaryotes, the exchange of hereditary information is quite a free-form, uninhibited activity. This all changed with the coming of the nucleus and the appearance of eukaryotes and putting genes in pairs. So it gets much more orderly. Now, why do we have genes in pairs? Why do we have chromosomes in pairs? So we can take those pairs apart at meiosis and put them back together again at fertilization. You could say, well, maybe we have genes in pairs so that geneticists can be employed. Otherwise, there'd be massive unemployment in our ranks. So this means we have an alternation of diploid and haploid phases. Anybody recognize that organism? Yeast, yeah, right. This is Saccharomyces, and this little thing over here, that's a tetrad. So actually, when I was in David Botstein's lab, my bench, I was the only person working on human genetics. My bench was right next to the tetrad dissection scope. So I have had tetrad envy for all of my, anyway. So yeast put the 4 products of meiosis in a single tidy little package, which has contributed enormously to the power of yeast genetics. Okay, so you have alternation of diploid and haploid phases. It turns out that meiosis looks to have been invented once. This was a dramatic invention on the order of 1.5 to 2 billion years ago. So this is when sexual reproduction as we know it came into being. But it turns out we got to pile another invention on top, which is 2 different kinds of gametes. So dimorphic gametes. And whoever makes, and we have a big one and a small one, and whoever makes the big one we call a female, and whoever makes the little one, we call a male. Now, I do not want to claim responsibility for a certain executive order that was issued recently that actually echoes. I just wondered, did they watch me give this lecture? I don't know. But this is the most broadly applicable definition across the animal kingdom. Do you make the big gamete or do you make the little gamete? And it turns out, I'm pointing out, actually in yeast, they make two kinds of spores. They are creatively called A and alpha. They are structurally indistinguishable. So actually it's not always the case that one gamete is big and one is little. Yeast does it just in this simple way with isomorphic gametes. But now having gametes of two different sizes was another big step. And it looks like this was invented several times by evolution, and in our lineage, on the order of 600 million years ago. And I will give as a poster species for taking sex to this level, I will offer up as a poster species the turtle. Now, why the turtle? I'm showing you here, and apologies for the explicit nature of the photograph on the top. If you consider this offensive, please just avert your gaze for a minute, and I'll tell you when you can look again then. So, but down below, I show line drawings of the internal anatomies, the reproductive anatomies of a male turtle and a female turtle. And they are every bit as different as the reproductive anatomies of a male and a female mammal. But the amazing thing about turtles is they don't have any sex chromosomes. Do you know how sex is determined in turtles? Shout it out. Somebody here, I'm sure, knows. Yes, temperature, right. So basically, it turns out that male and female turtles have identical genomes. They have identical genetics. It's just the temperature at which the egg incubates that determines which of these two anatomies you get. So the existence, I think if you like epigenetics, I think this is the most amazing example of epigenetics. Two different readings of the same genome give you two different sexual anatomies, two different reproductive anatomies. So the existence of two sexes is purely epigenetic. Males and females read the same genome differently. Okay, so the point I'm making is you can have meiosis. Both male and female turtles do meiosis. They have dimorphic gametes. One makes a big gamete, one makes a little one, but they haven't bothered to go on to the fourth invention, which is sex chromosomes, which can be, for example, XXXY, as in us and in fruit flies, for example, or ZZZW. Do you know any species that have ZZZW sex chromosomes? Birds, also butterflies. Not everything, you don't have to have wings necessarily to have ZNW, but, and it turns out that sex chromosomes have been invented, have evolved many, many, many times independently across the animal and even the plant kingdoms. But the real reason I'm putting this up here is to give you the sense, well, okay, if you read a chapter in a textbook on developmental biology, well, if you get a textbook on developmental biology, it'll have a chapter on sex determination. Where does that chapter on sex determination begin? Where does our model of how sex determination in humans or mammals work, where does it begin? It begins with the X and the Y chromosomes. What I'm trying to show you is that is not the way it came to be. So it is so basically, sex chromosomes are a late adornment to a perfectly functioning system of sex determination. We get it, we always teach it upside down. But I'm just trying to help you understand that the sex chromosomes are really a Johnny-come-lately in the world of sexual reproduction. Okay, so, and I'd say in mammals, in our lineage, our sex chromosomes began to emerge on the order of 200 million years ago. And I'm going to give you a little better understanding of how that all came to work. So I'm looking over here at Caleb and Alejandro. This is stuff I haven't taught you guys about yet, right? We have a lot of conversations about all this. So this is a new level. We got to take you through the evolutionary stuff. So where did the X and the Y chromosomes come from? Well, so now we're going to go, so we just said that birds are ZW and we're XY. It turns out that we have a common ancestor that existed 300 million years ago. This is a problem. How are you going to explain this? Shouldn't this be really conserved? Well, it turns out, so we have an XY system, which means, and this is really good to know next time you're at a cocktail party, you know, you can just casually ask somebody, hey, are you XX or XY? You know, ZW, whatever, wherever you want to go. But whenever a species has the two different sex chromosomes in the male, the naming convention is we call them X and Y. If the two different sex chromosomes are in a species are found in the female, the naming convention is we call them Z and W. Okay? It does not imply homology. So it turns out the Z and W in birds are not homologous to the Z and W in butterflies. They just share a naming convention. Okay, but you can see we got a common ancestor back here 300 million years. We got a problem. How are we going to explain how this came to be? So I'll just give you, I'm going to go right to the answer that we've, in the lab over the last 25 years, we've come to appreciate, come to reconstruct. And that is, it turns out the human X and Y evolved from ordinary autosomes that remain autosomes in birds. So this is the most amazing thing. So next time you go to a chicken farm and you have your automated DNA sequencer with you, you know, probably a device about like so, and you sequence the first chicken you encounter, and then you take out your smartphone, you do the alignment with your own genome sequence, you will find that your X chromosome sequence aligns very nicely with the short arm of your X will align with chicken chromosome 1, and the long arm of your X will align with chicken chromosome 4. Now, there's a few base pair substitutions that have arisen over time, but the alignment is unambiguous. And this actually, you know, provides pretty strong evidence of the autosomal origins. Where in the world then did the chicken Z and W, or actually we have some Canadians, so I should say Z and W. Okay, so where did Z and W come from? It turns out the chicken Z and W arose from other autosomes of our amniote ancestor that, guess what, that remain autosomes in us today. So next time you are contemplating your chromosome, your own chromosome 9, realize that it could have become a sex chromosome, as it did in birds. So, and we've actually gone on now to do these reconstructions in snakes, some of which are XY, some of which are ZW, and we find that even yet additional autosomes of the ancestral, of our amniote ancestor have become sex chromosomes. So, and one of the things along the way we thought, well, wouldn't it be cool to really reconstruct this evolution of the X and the Y from ordinary autosomes? And so over the years, we've reconstructed in silico the autosomal ancestors of the mammalian and the avian sex chromosomes, and now actually of the snake sex chromosomes. I'm just going to show you how this turned out in the case of our human X and Y chromosomes. So we're going to reconstruct this ancestral autosome that would become our X and Y, recognize that we have to reconstruct it in silico because this animal doesn't exist anymore. This was an animal that existed 300 million years ago, a common amniote ancestor. And basically, what we're able to determine, and a lot of this is really the work particularly of Winston Ballot, what he was able to reconstruct was that the chromosomes that gave rise to the human X and Y chromosomes carried about 639 genes. That's a pretty precise number because he was able to actually reconstruct it quite precisely. And it turns out that, so imagine now you're traveling back in time, 300 million years, and you're going to visit the origins of your own X and or Y chromosomes. And what he found was that the human X chromosome today retains 629 of the 639 ancestral autosomal genes. So the X chromosome has done a really good job of hanging on to. So the X chromosome is almost a copy of that ancestral autosome. Now, truth is a little fine point. It turns out the X chromosome has also gained a bunch of other genes, but I'm not going to say more about them. But it did a really good job of hanging on to the original genes. Now, we turn to the Y. The Y chromosome. I see a number of individuals, mostly without a Y chromosome, beginning to snicker at this point. So the Y chromosome, it turns out, retains only 17 of those 639 ancestral genes. Some are shaking, even speakers here are shaking their heads with, disdain at this. Okay, so only 17 of the 639 genes are preserved today. And this is actually, I show this as a true Venn diagram. So all of these 17 genes also survive today on the X. So this means 17 XY gene pairs that stem from an ancestral autosome that became the X and the Y. Okay, so so how'd this all work? So what we understand is that the human X and Y chromosomes evolved from ordinary autosomes. This actually ought to be the main story in the textbook now. I don't know when we'll be able to get to the place where we can teach our students that this is a powerful lesson in molecular evolution. But anyway, so I'll tell you that on an ordinary pair of autosomes, about on the order of 200 million years ago, we think that a mutation arose in a pre-existing gene to become what would live on today as the sex-determining gene SRY. I'm sure some of you have heard of this sex-determining gene SRY on the Y chromosome. And what we understand then is that in an increasingly expansive region, crossing over or free trade between the X and the Y was suppressed in a region by region basis. It was as if you'd put up a wall between two countries and suppressed trade there. So the exchange, and it turns out that as this goes on, the suppression of free trade results in the long-time loss of genes from the Y chromosome. So it basically is adopting isolationist strategies, and its economy decays, it loses genes. The analogies actually hold here reasonably well, to be honest. You may think I'm speaking facetiously, I'm not. And I'll just throw on, why does the X chromosome, here's a question for all of you, why is it that the X chromosome does not suffer the ravages of this suppression of crossing over and exchange with the Y? How is it that the X continues to keep its genes healthy? Because it is continuing to enjoy the rejuvenating benefits of exchange, where? In female meiosis, right? Between two X chromosomes. Okay, so So we end up with a Y chromosome that is much smaller and has limited trade with the X chromosome. And so we end up with this very small number of surviving genes. How many? 17. 17 surviving genes on the Y chromosome. And now we're gonna look at how this turns out. So it turns out there's this little green bit up at the top of the short arm of the human X and Y chromosomes, which is called the pseudoautosomal region. How many folks have heard of the pseudoautosomal region? It's a sort of familiar term. Okay, so the pseudoautosomal region is an area where crossing over between the X and the Y chromosomes is a normal and frequent event in male meiosis during spermatogenesis. So the X and Y chromosomes up there still remember that they came from autosomes and they swap bits, but it's a very small region. So down in the non-pseudoautosomal X, which I'll abbreviate as NPX, or the non-pseudoautosomal Y, NPY, we have these 17 surviving NPX, NPY gene pairs. And just to give you a sense of what that means, I'm picking out one of those XY gene pairs, KDM5C, KDM5D. These genes in the coding regions are 85% identical. They're 85% identical at the DNA sequence level. They encode proteins that are 87% identical at the amino acid sequence level. And it turns out that these genes, KDM5C, KDM5D as an example, they are both expressed in human tissues. And these two are both expressed quite widely, quite broadly across the body. So that means that XX tissues express exclusively the NPX, or X chromosomal isoform, and XY tissues express both the NPX and the NPY isoforms. So now you're starting to get the idea, my gosh, there are some now, as a result, some biochemical differences between XX and XY cells across the body. And most of these XY gene pairs have no known roles in sex differentiation as canonically or classically described, with one very notable exception, which is one of these surviving gene pairs is SRY and a homologue on the X chromosome called SOX3. So this SRY sex-determining gene did not come out of nowhere. It was on the ancestral autosome, and it became a sex-determining gene, and its SOX3 homologue on the X persists today. Okay, and it turns out that of these 17 NPX-NPY gene pairs, 7 of them are, as best we can tell at this point, 7 of these NPX-NPY gene pairs are expressed in every cell type in the body. Now, I've said in most cell types just to, provide a small insurance policy going forward, but and it turns out that these 7 NPX-NPY gene pairs that are expressed in virtually all, if not all, cell types encode central players in gene expression. So I just list the processes here. So chromatin modification, transcription, translation, protein stability, and degradation. So that actually these NPX and these surviving genes, these genes that survived on the Y chromosome and on the X, are not a random sampling of the genes that were on the ancestral autosome. They are the most central players of all those genes. And so these genes are collectively required for or regulate the expression of all protein coding genes in the genome. And so I will sort of wrap this up by talking about, I'm going to compare old understandings of the human X and Y with new understandings. And by old understandings, I mean what is still taught in most universities and medical schools around the world, but we'll bring it up to date here. So the old understanding was that the human Y chromosome functions only in the testes, in basically creating testes as opposed to ovaries and in spermatogenesis. And we understood that in female cells, the second X, the so-called XI, is silent. going to have much more to say about that in my second lecture. And so if you take these two things together, you realize that, the old understanding, and I would say this understanding still permeates the brains of most modern biologists because this is what we were all taught. It means that across most of the body, these two understandings translate into saying that both males and females were functionally XO across the body, right? If the Y is silent outside the testes, and if the second X is silent, so we were, and this provided a really powerful simplification of how to think about how sex differentiation works. Those were the old understandings, still, I would say, deeply embedded in the minds of most modern biologists. But what we now understand is actually that there are 7 different NPY genes that are expressed throughout the body. And these regulate expression of thousands of other genes. And there are hundreds of genes that are expressed from the so-called inactive X chromosome. I'll have much more to say about that in another hour or two. And it turns out those genes expressed from XI include NPX-specific versions of the NPY's broadly expressed global regulators. These are the NPX-NPY gene pairs that are widely expressed and are central players. So what this means is that throughout the body, that XX is not equal to XY at the most fundamental biochemical level. And I'll just wrap this up by saying, and apologies for the sort of gendered symbolism here. One of my hobbies now is to, as I travel around, to take photographs of the symbols outside bathrooms. And actually, this is the symbolism that I find most frequently on this campus, actually. Right? So I felt that I couldn't use any. No, I won't give you a hard time about that. So the prevailing view, the old view, is that whether you're XX or XY is of direct biological relevance only in the nether parts, only in the gonads. But what I'm telling you is that there are, in fact, biochemical differences that are the result of the evolution of the X and the Y from ordinary autosomes. There are subtle biochemical differences between XX and XY cells all across the body before they ever see the effect of circulating sex hormones, which were thought to be, in the old model, were thought to be the only source of masculinizing or feminizing signals outside the gonads. Now, I don't mean to throw away the old idea. I mean to actually hybridize it with this one. And so I will say whether you're XX or XY is of immense and disproportionate direct importance in determining the fate of the fetal or embryonic gonads. But then we've got, as the sex hormones emanate from the gonads, they wash across bodies that are already biochemically distinct by virtue of the evolutionary schemes that I've told you about. So that's all I wanted to say by way of background. And so I want to recognize lab members and alumni who reconstructed the evolution of the X and the Y chromosomes. A lot of what I'm telling you about is really the work of decades of trainees, including I'll point out, let's see. I'll point out one who is in the room here, Doug Vollrath. Let's give it up for Doug, and right, who actually played a big role in the mapping of the detailed mapping of the human Y chromosome that preceded the sequencing that made it possible to identify all these genes. And just for fun, that gave me the opportunity, since I hadn't seen Doug in a few years, gave me an opportunity to get out some old photographs. Doug is shaking his head. This is outside the Whitehead Institute. This is a photograph from circa 1990. And I want you to, let's see. You have to shout out when I get to Doug's head here. Hey. Okay, and since I don't get to do this that often, let's see. We used to take, that time we'd take a fair number of lab hikes in the White Mountains of New Hampshire. And here is one photograph of Doug together with Andrew Zinn. It was just a few years ago, Doug. So, all right. And with that, I want to thank all those who actually have made this work possible over the years. So I point out to HHMI, Whitehead, and the Britt Jepson Darbeloff Center on Women's Health, which is the name of our lab. And I'll mention other funding sources here that have contributed as well. And I will stop there. And let's see if, I don't know if we have any time for questions or so. Yeah, okay. Yeah. Okay, thank you very much. Right, yeah. Yeah.